During interphase, DNA synthesis occurs. This results in the formation of paired chromatids, and the centrioles also replicate.
Chromatids get condenser. Homologs smoothly pair along their lengths, aligned gene by length. At this point, crossing over is completed while homologs are held tightly together by proteins along their length. This process is called synapsis. To briefly explain, crossing over is exchange of corresponding segments of DNA molecules by non-sister chromatids. Synapsis ends in mid-prophase. Its homologous pair has one or more chiasmata points where crossing over occurred. Homologs are still associated due to cohesion between sister chromatids. Centrioles migrate to the opposite pole, and the nuclear membrane breaks down.
The chromosomes disentangle so that pairs of homologous pairs are arranged on the metaphase plate. One chromosome of each pair faces either pole. The microtubules are extended. Kinetochore microtubules are attached to one of two sister chromatids, while non-kinetochore microtubules stretch from pole to pole.
The first stage of anaphase in the meiotic cycle begins with the breakdown of proteins, allowing the homologs to separate. The homologs then move towards the opposite poles, dragged by the spindle apparatus. The chromatin cohesin continues at the centromere, causing the chromatid pairs to move together toward the same pole.
By now, each side of the cell has a complete set of replicated chromosomes. Chromatids include regions of non-sister DNA. The cell will divide at any time through cytokinesis, in which a cleavage furrow forms for the animal cells and a cell plate forms in plant cells.
This stage is typically called interkinesis, and DNA replication does not occur during this stage of interphase.
During prophase 2, spindle apicellars form. In late prophase 2, chromosomes, each still composed of two chromatids associated at the centromere, move towards the metaphase 2 plate. Centrioles move to opposite poles, and a chromosome appear to shorten and thicken.
The chromosomes are lined up single file on the metaphase plate in the center of the cell. Due to crossing over that occurs during meiosis 1, the two sister chromatids that make up each chromosome are genetically different. Again, the kinetochores of sister chromatids are joined to kinetochore microtubules that stretch from either pole.
During the second stage of anaphase and meiosis, the chromosomes are separated as a result of the breakdown of proteins between the sister chromatids. As individual chromosomes, the chromatids progress towards opposite ends of the cell. During the second stage of anaphase and meiosis, the chromosomes are separated as a result of the breakdown of proteins between the sister chromatids. As individual chromosomes, the chromatids progress towards opposite ends of the cell.
Nuclear membranes form around the chromosomes, and nucleoli reappear, while the chromatin decondenses. Each cell divides through cytokinesis. The resulting 4 daughter cells have haploid sets of chromosomes. These cells are known as gametes, which have half the amount of chromosomes as their somatic cell parents.